Hello. Today we'll be having our lecture number 6 in a series from Pathology by Robbins and Cotran, ninth edition. Today we'll discuss immune system diseases along with diseases of major histocompatibility complex and hypersensitivity diseases of complement. Immune system diseases. Immunity is a state of protection from infectious pathogens. There's innate immunity, which is natural or native, and this is always present. Adaptive is acquired immunity. Innate immunity components are common to all species of the same type. Epithelial barriers, leukocytes, and plasma proteins of complement systems. The innate cells recognize shared microbial determinants. They bind to toll-like receptors which recognize a set of molecules, and these activate leukocytes and interferon cytokines. They also activate inflammasomes, which generate interleukin-1, which is active in inflammation and antiviral defense. Adaptive immunity consists of lymphocytes and antibodies, humoral and cell-mediated lymphocyte subclasses. So among these are these broad categories where the antigen recognition, where the B lymphocyte microbe antibodies which respond to and they neutralize the microbe through phagocytosis, including complement activation. There are helper T lymphocytes, which have microbial antigen presented by antigen-presenting cells, and this causes cytokines which activate macrophages, increase inflammation, and their activation or proliferation and differentiation of T and B lymphocytes. Cytotoxic T lymphocytes, CTL, also have interactions with the infected cell presenting the microbial antigen. There's binding there, and there's direct killing of the infected cell. Regulatory T lymphocytes are involved in suppression of the immune response. And finally, there are natural killer cells, NK cells, and these have direct killing of the infected cell. Clonal selection is involved. Lymphocytes can utilize somatic recombination of receptor protein genes to recognize up to 1 billion antigenic determinants. The T lymphocytes, they're helper T lymphocytes, which stimulate B lymphocytes to produce antibodies and stimulate cytotoxic lymphocytes and regulatory T lymphocytes. The T cell receptor is linked to a complex which sends transduction signals. CD28 sends a cell-mediated response. This is a schematic of that. The antigen-presenting cell, there are class 2 MHC, major histocompatibility molecules, the alpha and beta chain. CD4 and CD28 are markers through CD80 within the membrane. There are CD3 proteins, epsilon chains, TCR heterodimers, and CD3 proteins, peptide antigen in the center, and these send signals through the CD4 cell recruitment, signal 1 and signal 2 through CD28. B lymphocytes have membrane-bound IgM and IgD antibodies. They recognize antigens which stimulate plasma cells and antibody formation. The CD21 recognizes the complement products. And here's an example of that. This is the antigen presenting, this is IgM, membrane bound. Complement protein is a cofactor through CD21. Again, you have the membrane bound Ig beta, Ig alpha, which sends a signal 1 and a signal 2 through CD21. Dendritic cells are present under the epithelial surface and in the interstitium. These capture the antigens and present them to B cells. Macrophages present phagocytosed microbial products to T cells. These are active in cell-mediated immunity, and these are cofactors in phagocytosis of antibody-coated, so-called opsonized microbes. Natural killer cells destroy virus-infected cells and tumor cells. No prior exposure is needed. This is a regulated response, and these are innate immunity cells. Lymphoid microarchitecture, you have lymphocytes produced in the thymus for T cells and bone marrow for B cells. Lymph nodes have an organized microenvironment which facilitates immune response, from dendritic presentation to lymphocyte differentiation. Other lymphoid organs exist, such as oral tonsillar tissue, spleen, and small intestine. The major histocompatibility complex, these are genes that are present on chromosome 6. They are highly polymorphic and they have two classes. Class 1 consists of molecules on platelets and nucleated cells. HLA-A, HLA-B, and HLA-C are derived from viral and tumor proteins within the host cell cytoplasm. These form complexes in endoplasmic reticulum. They're transported to the surface. They're recognized by CD8 lymphocytes, which induce an immune response. Class 2 antigens are HLA-D. These antigens are extracellular. They're transported in vesicles, associate with class 2 molecules, and then they are presented to the surface of the cell where CD4 lymphocyte recognizes the complex. MHC molecules are involved in cell-mediated immune response. We inherit immune propensities through the MHC locus and HLA haplotype. The correct immune response is garnered against cytoplasmic antigen, class 1, and extracellular forms that are internalized, and this is class 2. Cytokines induce inflammation. They inhibit virus replication. These are mainly produced by macrophages, dendritic cells, NK cells. CD4 lymphocytes produce cytokines in adaptive response. Other cytokines stimulate hematopoiesis, and these are colony stimulating factors. This is a schematic of this process. Again, you have the dendritic cell with the antigen. The antigen recognition in the lymphoid organ, which is here, the CD4 T cells, which produce CD4 effector T cells. These migrate into the circulatory system. There are also CD8 T cells, which stimulate CD8 T cells here through this interaction. And these effector T cells migrate to the site of the antigen. So CD4 effector T cells encounter phagocytes with ingested microbes. Cytokines are secreted. You get inflammation. You get macrophage activation, killing of ingested microbes. And you have also CD8 T cells, which emigrate. And these also interact with infected cells with microbes in the cytoplasm, and there's direct killing of infected cells. These are all cascade reactions, with naive T cells becoming activated through the IL-2R receptor, and you get proliferation of effector T cells and memory T cells. Another representation, there's a proliferation phase, differentiation phase, and a series of reactions, antibody secretion, class switching, affinity maturation, memory T cells, with a whole bunch of effects over here. So the microbe creates helper cells, interact with this, you get proliferation, you get differentiation, antibody secreting plasma cells, early phase of IgM, later phase of IgG. There's high affinity IgG involved in maturation, and also memory B cells, which will retain the means by which to produce a proliferative response. Afterwards, you get complement activation, and later on, with the phagocyte neutralization of microbes and toxins, opsonization or coating and phagocytosis, antibody-dependent cytotoxicity, lysis of microbes, and phagocytosis of opsonized microbes, and again, inflammatory response is mediated through complement activation. There are 4 types of hypersensitivity. This is an excessive and harmful immune response to an antigen. The classification, type 1 is intermediate. This is mediated by IgE and mast cells. They act on the vessel and smooth muscle. This is seen in allergies and asthma. Type 2 is antibody-mediated IgG and IgM antibodies. These injure cells. They induce inflammation. The prime example is autoimmune hemolytic anemia. Type 3 is immune complex, where IgG and IgM bind the antigen. There are deposits in tissue, and they induce inflammation. Systemic lupus erythematosus is the textbook example of this. And finally, type 4 is cell-mediated. They're sensitized lymphocytes, and this is seen in contact dermatitis and type 1 diabetes. Type 1 has late phase reactions where eosinophils are the primary cell involved. These are recruited by cytokines and is a component of a sustained reaction, as is seen in allergic asthma. Allergies are genetically determined. They're environmentally caused. It is not certain if increased early hygiene with later onset of disease is a factor. Systemic anaphylaxis is a situation of systemic shock, edema, and difficulty breathing. Immediate hypersensitivity examples are here. You can get anaphylactic shock caused by drugs, bee stings, food, which is manifested by hypotension caused by vascular dilatation, airway obstruction due to laryngeal edema. Bronchial asthma is another example where you have bronchial smooth muscle hyperactivity with airway obstruction and a late phase reaction with tissue injury. Allergic rhinitis and sinusitis is hay fever, increased mucus secretion, inflammation of upper airways. Food allergies or increased peristalsis due to contraction of intestinal muscles. Type 2 antibody-mediated hypersensitivity is where cells are destroyed by an antigen-antibody complex on the cell surface. Opsonized cells activate complement system. Another mechanism is antibody-dependent cellular toxicity or cytotoxicity. This employs NK cells and macrophages. This mechanism is characteristic of vascular rejection, and in some cases cell dysfunction occurs instead of overt destruction. Here are examples of antibody-mediated diseases: autoimmune hemolytic anemia. Target antigen is a red cell membrane protein. You get opsonization and phagocytosis, and the clinical manifestation are hemolysis and anemia. Autoimmune thrombocytopenic purpura, pemphigus vulgaris, vasculitis caused by antinuclear cytotoxic antibodies, Goodpaster's syndrome, which has lung and renal manifestations, acute rheumatic fever, which is a sequelae of an acute streptococcus infection, myasthenia gravis, which is acetylcholine receptor, which manifests itself with muscle weakness. Graves' disease, which is a form of hyperthyroidism, is a pathologic entity with the target being the thyroid-stimulating hormone receptor and antibody-mediated stimulation of TSH receptors. Insulin-resistant diabetes is manifested at the insulin receptor with hyperglycemia and ketoacidosis, and pernicious anemia is involved with the intrinsic factor of gastric parietal cells, and this entity is neutralized. You get decreased absorption of vitamin B12 and consequently a macrocytic anemia. Type 3 is immune complex mediated. This is an antigen-antibody complex that elicits damage at sites of deposition. The inflammation targets tissue, and this varies by disease. Here are examples of that. There's systemic lupus erythematosus, nuclear antigens, which are circulated or planted in the kidney. You get nephritis, skin lesions, arthritis, others. Post-streptococcal glomerulonephritis, streptococcal cell wall antigens can be planted in the glomerular basement membrane. Polyarteritis nodosa, which involves hepatitis B virus changed antigens. This is not always common. You get systemic vasculitis from that. Reactive arthritis, serum sickness, and arthus reaction, which is seen in vitro with various foreign proteins, and you get cutaneous vasculitis from that. Type 4 is T-cell mediated. It's mediated by cytokines from CD4 T cells and cell killing by CD8 T cells. This is the basis of autoimmune diseases. The prototype is delayed type hypersensitivity, where an antigen introduced causes a response in 24 to 48 hours mediated by macrophages. One prime example of this is a tuberculin reaction. The stages consist of CD4 T cell inflammation. The dendritic cell displays products. This induces cytokines, which differentiate T cells. The repeat exposure of the antigen causes cytokine release, and this induces macrophages to augment phagocytosis and amplify the inflammatory response. And we will stop there. Thank you very much.